Thank you. And as a last talk of this workshop, I wanted to thank IPAM for hosting us for this really great workshop. I really enjoyed it. Also, I would like to thank our attendees that came for this workshop and also my fellow organizers. I think it went out pretty well. So thanks, everyone. So as the last talk, I was also tasked with summarizing and kind of like giving also a segue to discussion. So I'll try to do it. I think you will see the themes that appeared towards the second half of this workshop in my talk. I will focus mostly on our work because that's where I have expertise. And please feel free to chime in. All right, so I would like to talk about in general, how do we start with the connectomics and go to dynamics and kind of go through this step of starting with static connectome and then adding dynamics, adding simulations to it, adding responses, and then what are the functionalities, organization that we see as a result of this connectome. And then eventually our goal is to get to behavior. And as I hope to show also back to kind of the structure, although this is really the kind of closing the loop question. I will be mostly focusing on model organisms and mostly focusing on C. elegans. And the reason for that is that C. elegans is, pretty simple model organism. As you can see, this is a worm, and also kind of a stepping stone towards more detailed organisms, those that where their connectomes are being resolved or already resolved right now. So in terms of C. elegans, why to focus on it is that it is pretty tractable nervous system, about 300 neurons, 302. And a lot of information has been gathered over the years, over more than 30 years. In terms of mapping the genome, in terms of the somatic connectome, this is the connectome that we'll be working with, 279 neurons, about 8,000 connections, and these connections are also known in terms of their types. Gap connections was mentioned here in terms of synaptic connections, chemical connections, which type they are, negative or like inhibitory. Most cases, in some cases, GABA is not inhibitory. There's always kind of like out of regular cases, cholinergic, glutamatergic, and so on. It is being constantly updated and more data that we are getting. And also additional connections are known, so beyond even these pretty standard types like neuromodulators and so on. So it's really interesting to work with it, and most of the information is available actually online in terms of atlases, like the worm atlas will include a lot of information, and if you are interested, feel free to go there and learn about the neurons. As Elad was mentioning, neurons are labeled, they have names, all neurons have names, and experimentalists usually kind of like know them really well and kind of even say, I work with this particular neuron, this is my work, basically. Yeah, great. So this is pretty much like a visualization of the connectome matrix. This was compiled by Varshny et al. From the different experiments in terms of organizing the connections into a matrix. And then the two colors that you will see here, these are the different connection types. I forget which ones are which. I think the blue ones are the gap connections, and the red ones are the chemical connections. Okay? So one could look at it as a graph, right, as a adjacency matrix. But And this is indeed kind of like a first step to look at it. But we already see by this coloring that actually it includes more than just like a single graph. First of all, like it's a directed, weighted set of graphs, right? Per each connection, we can think of another matrix that will be kind of joining here. And if we are thinking of another, like let's say we know how neuromodulators are connecting between different neurons, this will be another layer here, and so on, so on. Okay. So this is really great, and many graph theoretical methods were applied to this kind of representation and organization in terms of community detection, in terms of graph traversals, pathways, and so on. So really a central model. What we wanted to kind of think about, and inspired by the work by Nancy Coppell et al. That was published in Neuron, is to think of connectomes not as graphs, but actually as dynamic connectomes. What do I mean by that? And I think it was mentioned in one of the discussion topics is, let's invert maybe the graphs and consider connections as nodes, right? Because graphs are kind of nodes are static. Here, we actually need to consider that nodes are neurons, right? So they are dynamic. They will receive input into them. And edges are also dynamic. These are the connections. So chemical connections will be very different than electrical connections. So if we think of the connectome as kind of like our basic layer, on top of it, we will consider the dynamics. And this is the name that Nancy Coppell and the collaborators coined for this combination, dynome. So let's consider actually dynamical systems on top of the connectome structure. Okay, so that would be my main focus is how do we consider dynamical systems on top of a connectomic structure. So it's quite a big dynamical system, since now we have all these variables, right? And we have also these different types of, let's say, variables that we will define per each neuron. So it becomes like a high-dimensional system already by just like considering this definition. Okay. So inspired by that work, this is what we started to consider in 2014 with James Kunert, our joint student with Nathan Kutz. And what we proposed is to model the neurons in terms of their dynamics. So here you can see it's kind of like proposing to model a linear term for each neuron, and then input currents in terms of gap junctions and synaptic activity. There is no nonlinearity here, but nonlinearity appears in the synaptic connections, and we chose this nonlinearity to be a sigmoid function. And that's pretty important because if you consider kind of ReLU, piecewise linear or linear, you won't really get the effects that we were observing in this model. So how we would go about kind of like fitting this model? Yeah, go ahead.
I mean, if I pick a sigmoid, I could do a piecewise linear approximation of the sigmoid, and I know that of course, in terms of dynamical systems, you'll lose some theorems and have to do other things. But I can make a piecewise linear approximation as close as I want. So you're telling me even that's not good enough, no matter what.
I'm saying that piecewise linear, just two.
Oh, okay.
Pieces.
Well, okay.
Yeah.
Okay. But more complicated.
So yeah, more complicated
okay
could work. So you need actually the threshold, otherwise
no,
the system.
Yeah, no, I'm not arguing against the threshold. I was thinking about just the approximation of nonlinear by piecewise linear. Okay.
Oh yeah.
Okay. Sure.
Yeah. So not
ReLU.
Not ReLU.
No, right.
Yeah.
No, okay, I see what you're saying.
Mm-hmm. So how do we go about kind of inserting the parameters into this system? So the parameters in terms of neuron connections would be those weights that I showed from Varshny's work, where they consider to count the number of synapses or in terms of gap junctions, and then they normalize them. Okay, so that's an approximation, but this is given to us by the Connectome. In terms of these dynamic parameters, so some of them are available from the literature, and we did some study in terms of literature search. And also we looked at IV response curves in terms of approximately fitting them with experimental ones versus our model. Okay. So the model wasn't fit in terms of the exact parameters, and that was indeed our intention because we kind of wanted to see what will be the outcome of the Connectome. Okay, so how do you work with this model? You basically, you can inject any stimulus that you want into the neurons. It could be any time-dependent function, and then to observe the dynamics.
Sorry, so which part comes from the Connectome again? Which variable?
So, yeah, these GIJs are coming from the Connectome. Okay?
And the value?
All the other are global parameters, and they're based on literature and based on types. Okay, so what also helps is that, so we were playing with this model and finding lots of interesting stuff I will show you. But what also helps is to kind of visualize and reduce the dimension of that graph in terms of like showing the dynamics. So we chose a force-directed representation of the graph, although today many more dimension reductions and visual representations than in previous lectures were shown. All these are relevant. And basically the idea is then to choose your set of neurons that you would like to stimulate, and then let's observe the dynamics. So here's an example of stimulating. Let me skip. So here's an example of stimulating this neuron here that actually corresponds in experiments to escape and moving forward, okay, from danger. And the way that we wanted to visualize those dynamics would be on top of the graph, we will make the nodes to breathe according to the membrane potential, okay, that is being resolved. Yeah, so these colors are just different types of neurons. And these colors that you will see now will correspond to the membrane potential of the simulator. So let's see. So injecting a constant current into PLML creates this kind of dynamics. So it's kind of hard to look at it as like a full network, but what you can see is that if you focus on particular neurons, you will see that they start to oscillate. Some neurons are idle. Some neurons are oscillating on a faster scale versus other neurons oscillating on a slower scale. So actually, oscillations would be really a typical type of dynamics in this model. Yeah.
And do you know in the real system which neurons and what dynamics?
Thank you. I will get to it. Yes. When we inject a current into another neuron, let's say ALML, by the way, it corresponds to backward escape from danger. This is kind of dynamics we would get in this model. Okay, so you can see it's pretty different, right? Oscillations still exist here if you follow different neurons, but of course, the set of neurons that are active and also their time scales are very different.
Are you injecting the current like constantly, or just like seeding it at the beginning and then let it spread, kind of?
So in this case, it's a constant kind of clamp, yeah. Okay, so this is really interesting, and we were puzzled about, like, okay, how do we make sense of all these simulations? You can inject current and then stop. Eventually, it will all go to quiescent state, right? Because there is no extra stimulation. What if you inject like time-dependent stimuli, or just like constant stimuli, like what would be the solutions that are possible by this model. So just for this example, I'll try to answer this question. Just for this example, what we were starting to see when we look at particular groups of neurons, so these are motor neurons, and then injecting the current into PLM that corresponds to forward movement, we would see those patterns and kind of like forming also in motor neurons. So this suggested to us that there is probably kind of a justification for seeking dimension reduction techniques that will apply to these kind of snapshots in time, and then analyzing them and seeing what are the dimensions of them, how do they, what type of actually dynamics do we get? Are these limit cycles? Are these more complex movements and so on? So indeed, we did that. As phrasing Sarah, we were linearly smart only, and that's all we needed, principal component analysis on this type of data, whether all neurons or just the motor neurons. Motor neurons would be kind of cleaner, and what we would get is actually, as you can see here, the singular values. Only 2 are dominant. Basically, all the rest are almost 0. So this constant stimulation creates a dynamic movement, and this dynamic movement follows this very nice limit cycle in a 2-dimensional space projected onto principal components. You can also then find the modes, of course, of like which neurons are kind of, what are these modes in terms of the neurons and so on. So this led us thinking, and that's the title of previous paper is low dimensionality. Something that we observed by many these kind of stimulations is that we get low dimensionality, approximately 3-4 principal component modes, even for combined stimulations and so on, will give us an interesting projection. We could also then use this representation, PCA space, to study ablations in the model. So we would kind of mimic experiments. We would go to the network, ablate a particular neuron. Here, 2 neurons are ablated each at a time, AVA versus AVB, and then to see whether we get some change in terms of the original dynamics with no ablations. As you can see here, we are getting almost the same limit cycle in the case of AVA ablation, and quite different limit cycle. Actually, it's almost like the second mode disappears. We only have 1 mode in the case of AVB ablation. And some of those results are actually corresponding. In particular, this result is corresponding to the experimental findings that AVB neuron is actually associated. This is an interneuron that allows the pathway from PLM neuron, this neuron that we excited, to the motor neurons. Yeah.
So, sorry if you said it and I missed it, but first, if you added noise to either the synaptic connections or to the neurons themselves, how much noise in some qualitative manner you need to add to mess things up completely? And the other is, did you also look at what happens if there's no stimulus? Is there any notion of ongoing activity here that you...
Okay. Yeah, so in terms of noise, we did verify in terms of noise, both in parameters, and it turned out to be pretty robust, and also in terms of stimulation. Again, yeah, it seems to be pretty robust, although as I will describe, there are some sensitive and bifurcations. These are the points where you won't have robustness. And I will also touch upon that when we kind of build more dynamical components to the simulation. The second question?
The ongoing, if there's no stimulus, is there...
If there is no stimulus, okay, so this model, as it is right now, we will enhance it, but when you don't stimulate, the model converges back to the quiescent state, okay? Yeah, so this was really cool. So first thing from dynamical systems, since we are familiar with dynamical systems methods, and we have this low dimensional space, right? So like now I'm showing the low dimensional space in 2 modes and then time. Let's see if this solution will persist for any stimulus, right? So let's vary the stimulus. Let's do bifurcation analysis. And here is an example of bifurcation study that these attractors are not necessarily only limit cycles. They could bifurcate, and we can detect these bifurcations. We can use continuation in dynamical systems because we have this low dimensional space, and things are two-dimensional, as Mason would like things to be, right? It will become more complicated, but two-dimensional. We can do bifurcation analysis, at least numerically. So here are several examples. We actually continued and expanded on bifurcation analysis. We found a whole bunch of bifurcations and different states and multi-stability and bistability in the system, indicating those transitions that could happen when varying the different stimuli. Then, and this is just to show how far you can go with the low dimensional representation. This low dimensional space allowed us also to ask then, what if we just really apply many stimuli to our network and then collect all these stimuli into a huge matrix and then SVD this matrix basically. So this is the result that is shown here. And then we would go to the low dimensional space and then classify the different attractors into fixed points and limit cycles. And this sea of points, by the way, shown here as Tisney, but maybe other representations would be much more informative. So in this whole cloud of points, all these are fixed points. And then we get a very nice separation between fixed points and oscillatory movements and oscillatory, actually, dynamics. Okay.
So maybe just trying to understand what exactly you're doing with the stimulation. So are you stimulating the sensory neurons? Or are you stimulating anywhere? Are you stimulating one at a time? Or
here, all neurons are stimulated.
And okay, all one by one?
Yeah, one by one.
And why not?
Although one could do like a different setup.
And do you divide it at all between, like if you looked at the sensory stimulations, do you see something different? You get the kind of motor patterns you expect?
So that's interesting. Like for example, this PLM neuron you can see here, and it's kind of like the obvious neuron to stimulate, right? But you also will get neurons that are interneurons, right? So stimulation into them will also cause a particular.
Did you catch the RIM thing that, I guess, there's a story with RIM, right? Like the variability. I don't know if you're going to talk about it or.
There's many stories.
Okay, but the specific one I'm thinking of is like that it injects noise and produces individual variability and has a continuous.
Yeah, okay. We can take a look at it. I think RIM is definitely one of those neurons that appears. The interesting thing here is that you can also dive into these different attractors and see how different they are as well, right? So visualization plus this study of sweeping through different parameters, which are stimulations, provides you with organization. So if you don't know, it is a lot is known about C. elegans, but if you don't know, like these will be the target neurons that you would then target after this kind of analysis. And we did a similar process in terms of development. It was interesting to see that some neurons would be born as oscillatory kind of neurons or kind of creating oscillations, and then will become more fixed versus other neurons that will be as fixed point neurons and then will become oscillating neurons.
Wait, so was the input oscillatory or the input is?
The input is a constant. Yeah, but one could also apply, like, for example, stochastic input and then record. Yeah. All right, so with all these findings, it was really exciting to think about, okay, can we go to the next step of, all these oscillations are actually behavioral, and what do they mean in terms of worm movements, in terms of worm dynamics? So we are still working on this model. So one version of this model is published here, and now I think we are at the stage of really letting it to be explored further by the broad community. But the idea was to connect the nervous system to add more dynamic layers to it up to behavior. Basically to connect the nervous system with muscles and then muscles with a biomechanical model of segments that can move. So it's basically more ODEs that are interacting with these ODEs here. And not only forward integration, so integration to the body, but also incorporating the forces that it will act on the environment, which are very important, and then going back and seeing what's the effect on neural dynamics, what would be the feedback on neural dynamics. And we are benefiting from the knowledge of these maps that are known for C. elegans. Okay. So let me show, kind of revisit this scenario that I showed previously, PLM excitation, and then kind of a more complex scenario where the worm, after being excited with PLM, is also getting a short stimulus of ALM, and it is supposed to change its direction from moving forward to moving backward. So an avoidance scenario. So that's the outcome of the simulation. As you can see, now it got a stimulus and then it crawls backward. I chose this scenario because it kind of incorporates many of the dynamics that I've showed you before, because it shows that, first of all, the worm does take this coherent structure of crawling forward, crawling backward, although not perfect. If you will show it to experimentalists, they would usually say it's not a real worm. You need to kind of smooth everything here, where we would say, of course, but it's a first order approximation. Yeah.
Can you say something about what data were used to construct the model, and what's, and then this is, I guess, a prediction of the model.
Yeah.
So was it fit to behavior, or?
No, there was no fitting to behavior at all.
The neural activity just comes from
Yeah.
those equations you put down.
Yeah.
You had measurements of the connectivity.
Yes.
Any other measurements you use, like
so here you have
like
muscles, muscles map, right, and also measurements of kind of some parameters here in terms of the fluid dynamics and so on.
I see. And then in the neural network, there were a bunch of other parameters, right? Like the leak conductances and reversal potential.
Yeah.
And things like that.
So these are global parameters.
Where did they come from?
These are global parameters, and they were measured in experiments.
Okay.
And for each cell type, and that's what we are using.
And you know the signs of the.
For most of them, for some of them, it's still questionable. Like experimentalists would kind of argue whether it's negative or positive, and so on. We would have to decide. So something was assigned.
And then now you have the muscles, and the model for going from muscle activation to the kinematics, like what you see there. That how does that come about? That's also just sort of.
Yeah, integrating all this information through a muscle map that is known, okay, and then fluid properties that are also like usual environment for the worm, integrated here, and then all of this integrated together. So we specifically don't fit to behaviors or try to kind of overfit, because we are trying to see whether these patterns are kind of emerging or would be part of our fitting. Yeah.
Just a quick question. There's some old work from the 80s, so like by Holmes and Rand, this biomechanics only coupled oscillators. Is that the component that you're using? Or doing this
This
really
is,
from
yes,
scratch?
yes.
You're using
Yeah.
their old model for that part of it.
Macmillan Holmes, this is the model.
Okay.
Yeah. There's no citation here.
No,
but,
I was just wondering whether you were doing something different or just.
No, dashpots and.
Okay, great, thank you.
Yeah, thank you for mentioning. The really interesting thing, and I will get back to it, is that when we revisit the projection onto a low-dimensional space in terms of principal components, we get a bit different modes. And actually, the behavioral connection, this closing the loop, allows us to interpret these limit cycles in a better way. They are also arranged in a more sensible, interpretable way in the space. You can see kind of like the forward limit cycle that is more stretched, since the forward movement is indeed like more stretched, versus the backward one is almost orthogonal to it. And then the turn is connecting between these two limit cycles.
So what are the dimensions of this phase?
These are the first three principal components projection.
Okay, of the.
But integration of like the whole body model is integrated along with the neural dynamics. Okay, so the message here is that eventually it will also shape the phase space. You cannot just integrate the nervous system without taking into account the body and the muscles and all other dynamical processes. This will kind of the picture that we had previously was. Pretty good, but this one refines it and makes it more interpretable. But there's more work to do, and I would like to focus in my next actually slides, extensions, talking about neurobehavioral phase spaces and so on. But before that, I just wanted to show that this allows for lots of studies in terms of really diving into the circuits and neural dynamics in terms of trying different, seeing different paths and whether these paths are stable and incorporating different ablations and so on. So here is a demonstration of really kind of, I don't know why it went on to kind of including the previous circuit diagram. But we are investigating this circuit diagram and seeing that ablation of this AYZ neuron actually doesn't change the movement. This is the movement without ablation, and you can see that the worm is following it. This allowed us to, here is your, I think that's your neuron, right, Vivek? We are seeing that actually when you ablate this neuron, there is another alternative path that one could find by following and making additional ablations. And when all these neurons are ablated, then you will get that the movement will stop. Okay. And eventually, like how is it related to those small changes, right, and the sensitivity to the environment? I've shown you almost like a picture of something that is kind of like inscribed in the worm, and all these patterns will appear no matter what kind of stimulation we will inject. So this simulation that you will see here, this is where we, why is it slow? Yeah, it's kind of heavy video. But in this simulation, we are simulating, I think, 10 worms that are starting with the same initial condition, with the same kind of initiation. But then each initiation is a little bit different with a little bit of noise. And you can see that their movement is pretty similar to each other. You actually cannot see it because my computer is too slow. But the average worm will follow this red trajectory, while each individual worm will kind of go in its own way and will kind of detract from the path.
And in the model, there would never be spontaneous changes between different behaviors, or stopping once you start moving.
So in the model, when you're near the bifurcation point, this could happen, but the low-dimensional representation is robust to these changes. Yeah. Okay, so with this model, this allows for lots of exploration and trying to both dive into the circuitry of the nervous system and also to look at eventually when you compile it all together, how does it look? It really inspires novel methods in terms of understanding and analyzing the system. So I wanted to propose or to talk as much time I will have. So how much? Okay, 30 minutes. I wanted to touch upon these extension topics, what we can do in terms of neural representation space, in terms of dynamical system metrics that we can study such systems, and eventually also extending the model to be more accurate.
Did you answer Sandra's question yet, or is that coming? About your model makes predictions of neural activity. Do those match experiment, or?
Yeah, so I think
I must
kind of with these simulations, I think I showed that these match pretty closely. Yeah, up to some extent. Of course, they won't match exactly. You have the spontaneous behavior aspect, but in terms of both reproducing scenarios, and I just didn't include the videos of more scenarios that we studied due to time, but yeah, you would see pretty reasonable correspondence. All right, so in terms of neural representations, we were lucky here, and indeed, a linear dimension reduction worked, but I wanted to talk about, in general, and I think it was mentioned here today, in terms of how would you find this optimal representation space for these dynamic components. It could be dynamic neural dynamics. It could be also behavioral dynamics. It could be just in general time series. So we are drawing a lot of, and I think the literature as well, a lot of inspiration from this work on sequence-to-sequence learning with neural networks that was published in NeurIPS 2014. And the problem that they considered is translation, right? So having one text or sequence, and then you would like to translate it to another. Actually, this concept could be applied to many time series. It could be applied even to like toy examples, and that's what we considered. And the main structure that is important here is to have an encoder and then latent representation, latent state vector, and then a decoder, right? And the basic setup is to create a reconstruction task in terms of reconstructing the outputs that will match the inputs. Okay? So the network, what it does during this process, during training to reconstruct the inputs, it's learning within this vector to represent and to find the features that will correspond best to the data. And now this is an example. I will go really quickly over this example, but it actually shows that what the encoder is doing is to take trajectories that are starting from the same point, because the encoder doesn't have an idea what kind of input it will get, and then gets those trajectories separated as much as possible. And you can show that it kind of rejects these trajectories or has contrast between the different trajectories that are given to the encoder. And then the job of the decoder is to complete the trajectories after the encoder. And here you can see like a really simple example. How do we separate a circle from an ellipse that are projected kind of like on a 2D plane in a 3D coordinate space? And you can see what is the phenomenon that happens is that in the encoder, they both start from the same point, and after training, they eventually separate into two separable trajectories. And then the decoder states will take this information and kind of continue these trajectories to be eventually those closed trajectories. And then the linear layer from the decoder will project it back to a two-dimensional, to a plane. Okay. So this structure kind of could replace principal component analysis. This will be a nonlinear dimension reduction that will allow us, by using this methodology, if effective, to separate trajectories and to organize them in the phase space. We looked at this scenario in terms of joint body movements and classifying them into actions in this work in CVPR. We call this predict and cluster. And the basic idea is to use this encoder-decoder system and then to organize the time series of joints. The main point here is that we saw that it would be much more optimal if the input into the decoder will be a fixed state, which means that at each point of the decoder, the latent space vector will be given to the decoder to predict the trajectory from that point only. Which means that the decoder task is now not to predict only from the last point where it got the time series, but actually at each time step to project forward as many time steps as it needs till the end of the trajectory.
So what is the cluster part?
Are
you getting to the
yes, so
predict and cluster?
Yeah, so that's the predict part. So actually through this reconstruction task, but eventually what it means is that you predict for different time steps within the decoder versus reconstructing. You will construct this latent representation that would be more optimal. So instead of doing this with encoder-decoder, eventually you will get such trajectories which are better clustered in the space. So that's the clustering part. We call this weak training because the decoder kind of gets weak information, eventually doesn't allow, we don't allow to the decoder to kind of propagate, and it makes even the error in terms of reconstruction higher, but the organization in the latent representation is more effective. Okay, so this is a process of disentanglement, and I wanted to mention that similar ideas or similar decoders are also used for neural time series, and this is an example of LFADS, latent factor analysis, for example, where the authors considered spiking data. They didn't consider the weak training, but it's a similar structure in terms of eventually encoding and decoding the information. And the really interesting thing is that you can then relate between also neural latent space and behavioral latent space, and that's something that these nonlinear projections would be needed in order to do those joint representations. I wanted to mention our latest work on representations, and this was just published in NeurIPS, where we also started to think about how do we go back to the graph, to the structure with the neural dynamics. And in this case, we are proposing that for a given signal, we can propose a graph neural network, and I think it was mentioned here, which will have two parts. It will have additive objects or encoders and multiplicative encoders. And then this network will not just reconstruct the signal, but will also learn the graph structure, okay? And these adjacency matrices then can be used for analysis, right? Or if we don't know the structure, or completing the signal, but also to arrive to a more organized latent representation. And these are just examples how predicting, and again, our focus here is to say that it's not just reconstructing, you actually need to predict several steps forward in order for this network to be applicable. And the interesting thing is that if you look at principal components versus the embedding within this graph neural network, you will see that these embeddings are organized by themselves through the optimization of the structure or learning the structure. The network will organize the projections onto the embedding space. Okay.
I did have one question. The multiplicative unit, is it actually, so neural networks usually have
Right.
a bunch of addition and then a nonlinearity. So is there like a multiplicative, like pi sigma network?
Yeah, so we included the multiplicative part as kind of like a gain modulation effect. And it played a pretty significant role. I just wanted to mention that there is no limit of considering just the dynamics themselves. Once you have the dynamics and you are able to simulate or propagate your models, in this case, we are actually considering artificial neural networks. But we can use dynamical systems tools to further analyze and further construct latent representation. So in this example, we are considering Lyapunov exponents, which are characteristics of dynamics. So these will denote what kind of expansions and contractions we have in a dynamical system. And then analyzing these spectrum curves for different systems, okay? And this analysis could also be repeated for time-dependent kind of fashion, where you will get time series of these Lyapunov exponents called finite time Lyapunov exponents. So it makes sense to actually change your dynamic variables to Lyapunov to some other features if they make sense. As you can see here, maybe it could make sense because we are starting to see what are expanding directions, contracting directions, and so on. But even more effective would be to combine them with neural representation methods that I've described just now in terms of encoding them into low-dimensional space. Several examples that we were able to show in this work, it's on the archive and now in submission, is that we are able to kind of not know much about the parameters of our network. Here we are considering, for example, a random network that was trained to learn a particular target. And we are able to classify the different parameters within this network without even knowing them. Or in this case, we compared LSTMs, right? That's why it's in GRUs, RNNs, all these different types of RNN networks in terms of their optimality with respect to the sequential MNIST task. So the Lyapunov exponents by themselves, it's hard to interpret them. But once you encode them into a low-dimensional space, you would get an interpretation that seems to be clustered and organized.
Just so you understand what you're doing, so you're forming a time series of finite time Lyapunov exponents, and then you're embedding that time series into a low D space?
Yeah.
Okay. And that was the case both in the previous slide and on
So,
the slide?
yeah, there are two versions. One version would be to compute Lyapunov exponents for the whole propagation. So then you will get Yeah, you'll get spectrum, and then to look at this spectrum projected to low-dimensional space as a point here. So each point is a spectrum. Or to do finite time Lyapunov exponents, and then you will get time series, and then.
Okay. So like in the first case, you would train a bunch of systems, get
Yeah.
a spectrum for each
That's
system.
the first case.
ECAM.
Yeah.
Okay.
Yeah.
Um,
And I can answer more questions. Sandra is indicating that I have 0 time left. I will skip the electrophysiome because that's an attempt to enhance our model in terms of individual neuron dynamics that matter. I wanted to thank my students, group members, they are listed here, and collaborators, and also funding. And thank you for the attention.